for you today, right? Can you concentrate? No. Okay, all of you can stand. Please stand up. Stand up. Move your hands, limbs, everything. Okay. So this is my first lecture for you in this block. So here we'll be talking about one of the sections of anatomy that is called as embryology. Embryology means, what do you mean by embryology? Study of the embryo. Okay. So the fetus or embryo. Okay. So we have many divisions in anatomy. One of the division is the study of the development, the fetal development in the womb of the mother. Okay. So here today we will be talking about before this fetus is formed, how exactly the things are going on. You might have studied some of the things in different sections, and today we will talk about more about the development of the fetus, the initial developments. Okay. So, you should know that the development of the fetus in the first, second, and third week is very important. A lot of changes will be taking place. Okay. Okay. So you should know this. The development in the first week, second week, and third week, and this is very important, and it is common for all the development. Later, for each system, like the heart, development of heart is separate. Then the development of the lungs is different. The development of the intestine is different. The development of brain is different. Up to third week, it will be common together. Okay. Later, you will study the development of each different system. Okay. That is called as systemic embryology, where you study the development of one system. Now, initially, you will study in general the whole embryo, how it develops. Okay. So, to begin with, we'll talk about the fertilization, cleavage, and implantation in this lecture. So, these are the objectives for this lecture. Okay. So, we'll start from the fertilization. So, what do you understand by the name fertilization? What do you think? Fertilization.
Combination of sperm and ovum. Combination of sperm and ovum.
Okay. Okay.
You said it all.
Yeah. So it is, as you said, the fertilization is the fusion. Fusion. Okay. Joining of the two structures. One is called as the sperm, and the other one, the male gonad. You can call it as. It will be the sperm, which specifically, if you know, it will be the spermatozoon. And in case of the female gonad, it will be the ovum or the gamete. It will be similar. It will be gamete. Okay. So the ovum as well as the sperm, when they fuse together, that will be called as the fertilization. Okay. Fusion of the sperm and the ovum together. Okay, that is called as fertilization. But you should remember that these are haploid. Haploid. What do you mean by haploid? You know? What do you mean by diploid? Do you know the number of chromosomes in the body?
Yes.
How many?
46.
46. That is 44, and X and Y for the male, and 44 and 2 will be the sex chromosomes. Those will be, in case of female, it will be XX, and in case of male, it will be
XY.
Okay. So these are called as 23 pairs of chromosomes. But in case that is called as diploid. But these sperm and the ovum will have half of the chromosomes. So they are called as haploid. So they will have not 23 pair but just 23, not pairs. Okay. Yeah. So it will be just 23 chromosomes, not pair. So they are called as haploid. And once they join together, the sperm and the ovum, then they become diploid. Then that will be the actual number of chromosomes which will be formed in the body. Okay. So initially there will be the sperm as well as the ovum will be haploid in nature, half of the chromosomes. Once they fuse by fertilization, then they become diploid. The normal number of chromosomes is restored. Okay. So this is the fertilization, that is the fusion of the male and the female gametes. Okay. And this fertilization, where does it take place? In a structure called as ampulla. Ampulla. Where is ampulla?
Details.
Not in the uterus.
Hello.
Huh?
Hello.
Yeah, fallopian tube. Good. Okay. So this is the uterus here. Okay, this is the uterus called as harm, right? Huh? Yeah.
Yes.
Raham. Okay, raham. Okay. So okay, raham. Okay. So in Arabic. So this is the raham here, and this is the tube for that. This is called as the fallopian tube. This fallopian tube has different parts. 4 parts are there. Okay. One of the part, the largest part, dilated part is called as this is called as the ampulla. The most dilated part of the tube. Okay. Here is the ovary. If you see here in this picture, this is the ovary, this is the fallopian tube, and this is the uterus. Okay. So the ovary is releasing the ovum, and it will come to the ampullary part. And the sperm will be coming from here all the way to the journey. And here they will fuse together. That is called as fertilization. Okay, so this will usually the fertilization takes place in the fallopian tube in a particular part called as ampulla, the most dilated part of the fallopian tube, or it is also called as uterine tube, fallopian tube. Okay, and this fertilization will be studied under 3 stages. Okay, one is called as the approximation of gametes, how the 2 gametes, the male and the female gamete will approach, that is the sperm and the ovum come, approximation, then how they contact and how there will be fusion of the gametes, as well as the third is the what are the changes which are taking place, what are the effects of this fertilization. One is the approach of the 2 gametes, the male and female, the second is fusion, and the third is what are the effects, what are the changes which take place after the fusion. Okay. So these are the 3 headings under which we will study the fertilization. Okay. So the first is the approximation of the gamete, the male and the female gamete. Okay. As I said before, the sperm will be passing all the way through the vagina and then the cervix, and this is the uterus here, and it passes all the way up to the ampullary part, and the ovum will be coming from the ovary, release here and come into here, and here will be the fertilization. Okay, so the sperm and the ovum will come at the ampullary part, and then the deposition of the spermatozoa in the vagina leads to prostaglandin. Whenever the sperms are released into the vagina and the uterus, there is release of a secretion called as prostaglandin. Okay, and that leads to the contraction of the uterus, powerful contraction of the uterus. So once this semen is released, semen has the sperms, and within the semen there is prostaglandin. Whenever this prostaglandin is released, it will lead to contraction of the uterus in case of the female, and that will push the sperm upwards. Okay. Then also the coital reflex. So after the sexual act, this leads to a reflex within the vagina as well as the uterus, which again leads to a hormone called as the oxytocin, which again helps in the contraction of the uterus. Okay. So contraction of the uterus is the main thing which push the sperm. How they will come here, approach. The short duration here, if you see here, in this part, this ovum has a very short distance to be covered. It can easily pass. Okay, but what about the sperm? It has to go all the way, and it has to ascend upwards against the gravity, go all the way till here. So for all these things, it will be because of the hormone, the oxytocin, as well as the secretion of the prostaglandin, which will help in contraction of the uterus, and then it will push the sperm towards the ovum. Okay. This secretion is also mixed with the uterine glands. There will be uterine glands within the uterus. We'll study when we study the histology. At that time, they also release a lot of secretions. All these secretions with the important hormones and the enzymes, it will help in the pushing of the sperm into the ovum. But the sperm itself is motile. The sperm itself has the capacity to move. Okay, I will show you the next pictures where you will see how motile it is. You see here, in this picture, see, This is how the sperm will move. Okay. This is the side view, the above one, and this below one is the posterior view. So this is how this sperm will move.
So the contraction of the uterus will the sperm to move out.
Yeah, as well as the movement of the sperm. But the movement of the sperm itself has a very small role. The main will be the contraction of the uterus itself, which will pull the sperm upwards into the ampulla. Even though the sperm has the capacity to move, but as you can see here, the active motility of sperm does not have role or very small role. The main thing which will pull the sperm up to the ampulla will be the contraction of the uterus because of the prostaglandin as well as the oxytocin. Okay. The sperms are gradually reduced in number. Actually, when it is released, there will be almost 200 to 300 million sperms once there is sexual act and there is release of this semen into the vagina. When it is in the vagina, almost 200 to 300 million sperms will be there. Even if they are reduced to 100 million, then the person doesn't have children. Okay, it is called as oligospermia, where there is decrease in the number of sperm. How many sperms has to fertilize the ova?
200
to 300.
How many are released?
200
200 to 300. And even that comes to 100 million, no use. The person cannot have children. Okay, that is called as oligospermia, where there is decrease in sperm. So the number of released sperms will be 200 to 300 million sperms. But once they are passing through the uterus, see, this is the whole thing. Okay, so when this is the vagina, this is the cervix, this is the uterus, and this is the fallopian tubes, and this is the ovary. So the sperm has to pass all the way up to here. Okay, to the anterior part. When they are released here, there will be 200 to 300 million sperms. Okay, but there are many barriers through which the sperms have to pass through. Okay, so by the time it passes through from vagina into the cervix, this is the cervix. Okay, the cervix is very narrow here. Okay, so only 1%. Huh?
There is the contraction bit and the
Yeah. Everywhere there is contraction. That's why the sperms are pulled up. Okay. So by the time they pass from vagina into the cervix, they are reduced to 1% of the total number. What is the 1% of this one? 1% of 200 to 300 million is
20,000.
20,000?
200.
200,000? 200 to 300 million sperm. Out of that, 1% means how much? 100 million sperms.
2 million.
2
Huh?
million.
2 million. Yeah. So 200 to 3 million sperms will reach into the cervix. Okay, because there is this barrier. Okay. And even which they have passed, most of them die within 24 hours. Their life capacity is just 24 hours. Okay, most of them. Not all. Some have the capacity to be alive for almost 6 days or so. Okay. But most of these sperms will die within 24 hours. Okay. And the trip or the journey for the sperms to move from here up to the ampullary part will be taking from almost, the fastest will be 30 minutes, half an hour, up to 6 days. Okay, so it can be from 30 minutes up to 6 days. That is the whole journey they might take. Okay, some are fast, some are slow. Okay, and the oocyte is transported by ciliary beads as well as the rhythmic contraction of the uterine tubes. In case of the uterine tubes, again you study the histology where there are something called as the ciliary finger-like projections, which are called as cilia, which will be beating. Okay, which will be beating. They will pull up to here because of the contraction in the vagina and the cervix and uterus. It will be because of the contraction of these structures. But in the fallopian tube, not only contraction but also there are some finger-like projections which will be pulling the sperm, pushing the sperm into the ampullary part. These are called as the ciliary beads. Their beating is called as ciliary beads. Okay. As well as the contraction of the uterine tube itself. Okay. So the role of the movement of the sperm itself by itself is very small or negligible or not there. Okay. Now when it reaches, by the time it reaches the sperm, out of the 200 to 300 million, only 1 million or 2 million or 3 million will be reaching the cervix, right? And out of those, even most of them die, and by the time they reach the sperm, there will be almost 300 to 400 sperms. Out of these millions of sperms, only 300 to 400 will reach there, and out of these 300 to 400, only one will be penetrated. So why do you want so many sperms? What is necessary? Only one sperm can be done. Why do you need so many sperms?
To make competition.
Huh?
Still, that,
Pardon?
To destroy the wall of the ovule.
for
To destroy the wall of the ovule.
Okay.
To make sure that it's reached.
To make sure it reaches. Okay. Okay. All these might be the possibilities. One is the number, huge number is necessary. As I said, even if from 300 to 100 million, it becomes oligospermia. So that's why, as your friend said, all have some part. Okay. One is to make sure that it reaches, because most of them die. Okay. So we don't know. Only if there is only one sperm, it might die within 24 hours before it reaches. Right? So it has to be many. And why so much huge? We don't know. But there are many possibilities. Okay. Most of them will die, and they have to pass through so many barriers, and there are other barriers which will come across now. Okay. How would be contact and fusion? So contact of fusion of these gametes. Okay. So out of the, as I said, the millions of sperms, only 300 to 400 or 500 maximum of the sperms will reach the ovum. Okay. But among these, even 300, 400, 500, only one will penetrate. Okay. One of the reasons, as your friend said, why even we need 300 to 500, because all these other sperms will be helping in breaking some barriers. Okay. So by the release of an enzyme called as the hyaluronidase, okay, which will be released, and that will be breaking the barriers. So what are these barriers here? We'll talk about these barriers. There are three barriers which the sperms have to break before they enter the ovum. Okay, there are barriers apart from that. Okay, but these are the three barriers which are within the covering of the ovum, the female gamete. Okay, so there are three barriers. Which are those barriers? One is called as the corona radiata. If you see here the cells, this is the ovum here. So these are the cells here. They are present in the fashion that is called as corona radiata, because the cells are arranged in the fashion of radiation. Have you seen the sun and its radiations coming everywhere? So in the similar fashion, these cells are present in the fashion of radiation. So that's why it is called as corona radiata. All these cells which are around, they are present in the fashion of a chain, long chain, and they are in the fashion of a radiation coming from the sun. Just it looks like the sun and the radiation. Okay, so that's why it is called as the first barrier will be the cells which are around it. That is called as the corona radiata. The second barrier which they have to break is something called as the zona pellucida. There is a membrane here, thick membrane, very important membrane. Okay, which is called as the zona pellucida. And the third is pellucida. And the third is the deep inside. If you see in the ovum itself, there is a membrane here. This is called as the vitelline membrane. Vitelline membrane. And in between the zona pellucida and the vitelline membrane, there is a small space. This is called as the pre-vitelline space. Okay, there is a space here, so it is not a barrier exactly. So there is a space which is between the zona pellucida and the vitelline membrane. Okay. So these are three membranes which they have to penetrate. One is the corona radiata, the second is the zona pellucida, and the third is the vitelline membrane. So all these membranes are not easy to be broken down. So that's why we need so many sperms. Okay. So how do they break this barrier? Okay. So you should know, before we talk about the barriers, we should know that the sperms are released from the male into the female, right? Okay. The sperm will be maturing in case of the male itself before they are released. But the final maturation of the sperm will be taking place actually in the female once it is released. Okay. The sperms which are released from male into the female vagina are not mature, totally mature. The final maturation of the sperms will be taking place in the female vagina, and this is called as capacitation. Capacitation. Okay. Before fertilization, this has to take place. Before fertilization, they have to have the capacity to penetrate. So that final maturation of sperm is called as capacitation. That is the ability or the capacity for the sperm to penetrate the ovum. Okay. So before fertilization, the sperm has to undergo a process called as capacitation, followed by, then we have one more reaction that is called as the acrosomal reaction. We'll talk about that. Okay. So you should remember the sperms which are released from the male into the vagina, into the female, are not totally mature. Okay. But the final maturation of the sperm will be taking place actually in the vagina, or before penetration, or before the fertilization of the ovum. Okay, so that process is called as capacitation. So as I said before, capacitation is the last stage of maturation of the spermatozoa. Last stage of maturation of the spermatozoa. Okay, before it is released, there are many stages where the sperm will be becoming motile and other things. Okay, but the final or the last stage of maturation of the sperm, which will be taking place in the female genital tract, not in the male. Okay, once it is released, then it will be get the capacity to actually penetrate. Okay, and this requires almost 7 hours. It has to stay at least 7 hours in the vagina as well as in the other parts of the genital tract, the female genital tract, before it gets all this capacity to penetrate. Okay. Then we have something called as the acrosomal reaction. Okay, acrosomal reaction. If you see here, this is the ovum, and here is the sperm. This is the sperm head, and on the top of the sperm head there is something called as the acrosomal cap. So this is the sperm here penetrating, this is the ovum here. Okay. So similarly, if you see here, this is the tail of the sperm, and here this is the head. In the head, we have on the top of the head of the sperm, there is something called as the acrosome or acrosomal cap. Okay, so this acrosomal cap is very important. Okay, so just before approaching the oocyte, the capacitated sperm head. So the capacitation should have taken place. Okay, before it penetrates. Sperm head established multiple contact. If you see here, there are multiple cells. I will show you the next picture where you can see lot of sperms are all covering the ovum. Okay, and they have multiple contacts where they will be establishing multiple contacts between the plasma membrane. Here is the plasma membrane of this one. Okay, of the ovum as well as the outer membrane of the acrosomal cap. We'll talk about that. Okay, and then they release some very important secretions or enzymes. Acrosin as well as trypsin like chemical substances are released, which help in the breaking of these barriers. There are a lot of barriers here, so we have talked about the 3 barriers. Okay, so breaking of these barriers needs some release of some enzymes and other things. So when they are releasing and they are breaking this barrier, and these secretions are mainly released from the acrosome, that's why that is called as acrosomal reaction. Acrosomal reaction. This multiple contacts is known as acrosomal reaction when they release the secretion and break these barriers. Okay, that is called as the acrosomal reaction when they release the secretions from the acrosomal cap. Okay. See here in this video, you can actually appreciate how exactly it is taking place. Okay. See here, the acrosome is coming, the sperm head, and this is the acrosome, and it is releasing the secretions, and there is contact between the cytoplasm of both the sperm as well as the ovum, and now there are release of chemical substances which helps in the breaking of the barrier, and now the sperm is pulled inside the ovum. Once that acrosomal reaction takes place, then the only the sperm head, the tail will remain outside. Only the sperm head will enter. You can see here with the sperm head, which is very. Once again, if you can see here with the sperm approaching the ovum, and now there is contact between both the gametes, and now release of chemical substances.
then there is some reaction going on, which is called as the acromosal reaction. And now there will be calcium release, and now the cell, the head of the sperm is pulled inside. Okay, this is how it takes place. Yeah.
What's this, the green thing?
Which one? This is? Yeah, we'll talk about this. Here we can see here something called a cortical gland. They are important. We'll talk about that. Okay. The cells of the cumulus oophorus and coronary are moved by the liberation of the hyalinase. As a cell, there are cells, as we saw before in this picture. These are the cells of the coronary as well as the cumulus oophorus, a part of it where it is attached. Okay, all these cells will be moved out. First they have to be separated and pushed out. Okay, in the way of the sperm. So when you can see here, the sperm is approaching. It has to first move the cells here, which are in the coronary area. Okay. Then the sperm head will come in contact with the second membrane here. If you can see here, the sperm has come in contact with the zona pellucida. Okay, a thick striated membrane which has glycoproteins of ZP1, ZP2 as well as ZP3. Okay, all these three proteins will be there, which acts as the second barrier here. Okay, after the coronary radiata is pushed, now the second barrier is the zona pellucida, which has to be broken down. Okay. The sperm head binds with the ZP3 as well as ZP2 receptors and induce acromosal reaction. These are chemical substances, acrosin type enzymes which will be released and digest the zona pellucida. They will break this zona pellucida. Okay, this acrosomal reaction as well as the acrosome enzymes will release and break the zona pellucida. Okay. Now the sperm head will fuse with the vitelline membrane. If you see here, this is the vitelline membrane, which is nothing but the membrane of the ovum. Okay, so this is called as the vitelline membrane. Okay, so this vitelline membrane will be joining with the membrane of the acrosome itself, and now they will disintegrated peptides will be released, and now the head is released inside. Okay, after breaking of this barrier, the third barrier, now they will be entering. The head will enter without the tail. The tail remains behind. You can see here, the tail is outside and only the head will enter. During this process, as I said, there will be release of chemical substances as well as there is some changes within the calcium channels. Okay, channels. Okay, calcium wave will set in. So if you can see here, one sperm head will be entering into the sperm head. This is the acromosal cap, and now this is the acrosomal reaction, and it is releasing the chemical substances. Now once it comes with the contact with the vitelline membrane, now there is a release of the calcium channels or calcium wave, which will be very important. Okay. So the calcium wave sets in throughout the cytoplasm, resulting in important events in the fertilization. So why there is calcium release here? Okay, we'll talk about that. Okay. So the calcium release at the time of acrosomal reaction and entry of the head is very important because it leads to two important changes. One is it triggers the cortical granules in the periphery. If you see here, these are cortical granules which are there inside the vitelline membrane. Okay, once the sperm head enters these cortical granules, okay, they will all form a barrier now. Release of cortical granules causing the zona reaction, preventing the further entry of the sperm. As I said, there are 300 to 500 sperms which are surrounding. Okay, if one has entered, all others can enter. Once the barrier is broken, all can enter. So once the one sperm enters, then there is calcium wave and triggering of this cortical granules, and then there is formation of a second membrane barrier. Now it doesn't really allow the other sperms to enter. Once the sperm has entered, now they create a barrier. Suddenly there is a barrier formation, and no other sperm enters. So that will prevent something called as the polyspermia. If there are multiple sperms entering, then it's called as polyspermia. So it is prevented by this calcium channel, which will further helps in the cortical granules, which form a membrane around. Okay, and they prevent the entry of other sperms entering. So only one sperm will enter, even though there are 300 to 500 sperms, only one will be entering. Okay. And the second important change which will be taking place is, even as I said, the sperm is not mature till it is released into the vagina. Similarly, even the ovum is not completely mature. It is in the second meiotic division. Second meiotic division. If you have studied, or you will be studying, I don't know, you will be studying the divisions of and how the sperms and the ova are formed. Okay, spermatogenesis as well as the oogenesis. I don't know whether you have studied or not. Okay. In the final stages of the oogonium, the final stage is, before the final stage, it will be taking place in the second meiotic division. Okay, so this second meiotic division is not complete till the sperm head enters the ovum. Second meiotic division in the ovum is not complete till the sperm head is entered. So once the sperm head is entered, then there is calcium channel wave, and then the second meiotic division is completed. Now the sperm is mature as well as the ovum is mature. Okay, did you get it? Sperm, as I said, will mature once it is released into the female genital tract. The ovum will be mature, final maturation of this ovum, that is the completion of the second meiotic division, will be taking place once the sperm head enters and there is a calcium wave channel. Okay, okay. So these are the two important things which will take place because of the calcium wave which will be occurring once there is acrosomal reaction. Okay. As I said before, both are haploid, that is, they have half of the number of chromosomes, the sperm as well as the ovum. Now with the fusion, what happened? Now they become diploid. Now the actual number of chromosomes are restored. Okay, so if you can see here, so this is called as the pronucleus, female pronucleus with the male pronucleus, with the head of the sperm. See now here, the sperms are still there, but they are just outside, they cannot enter because there is a barrier formed with the cortical granules. So no other sperm will enter. Okay, so only they will be outside, they cannot enter. Only one has entered with the tail outside, this is the head of that sperm. This is called as the male nucleus, or it is also called as the male pronucleus. Okay, and this is called as the female pronucleus. After the second meiotic division and the release of the second polar body, this is called as the second, this is the female pronucleus. Now the male and the female pronucleuses are there. Now the diploid number of chromosomes, the actual number of chromosomes have achieved. Now they join and fuse, and now they start dividing. Okay.
Female nucleus or pronucleus?
Pronucleus. It is called as pronucleus because it is totally mature. Once there is fusion, then they will start, the actual nucleus will form, and then they start dividing. Okay. So, as I said, the DNA will start replicating now. Okay. So, after this joining of both the male and the female pronucleus, now the actual number of chromosomes are restored, and now the DNA will start replicating. And now, if you can see here, this is the male pronucleus, this is the female pronucleus. Now, the DNA starts replicating, and then they start dividing, and they divide into multiple cells. So there was only one cell initially. So this is the cell. Okay, now it starts dividing. You can see here, this is the male pronucleus, female, and now they are joined, and now cells division start taking place. Okay, now with the duplication of the DNA, so this is how the head of the sperm entering. Now it becomes pronucleus, this is the second female. Now there is division.
It's called the pronucleus.
Pardon?
Called the pronucleus.
Pronucleus.
That's it. The female.
The male and the female, both are called as female pronucleus, male pronucleus. Okay. Here in this picture they have mentioned nucleus, but usually it is called as pronucleus. Male pronucleus, female pronucleus, both will join together. Okay. Now the actual number of chromosomes are restored. Now the DNA starts replicating, and then there will be divisions. We'll talk about that. Okay. So here you can see this is the electronic microscopic picture. This is the actual picture. Here you can see the male pronucleus as well as the female pronucleus within a single cell. So this is a single cell stage of the human. Okay. So this is the picture again. Once again, you are showing all the features. Okay. So here, if you can see here, this is the whole of the ovum, and here you can see the coronal area. This is the zona pellucida, and this is in the second stage of the meiosis. Second stage of the meiosis. It is not completely mature. Once the sperm head enters, they have shown the whole sperm, but only the head, as I said, head will enter, the tail will not be there, it will be outside. Okay. Now once the sperm head enters, then there is completion of the second meiotic division. Now this becomes the female pronucleus. Now the entered head, this is called as the male pronucleus. After the second completion of the second meiotic division, there is release of the second polar body. With every meiosis, there will be release of a polar body. With the second completion of the second meiotic division, the second polar body release. Okay, now the male and female pronucleus are there. Now they start fusing with each other. Okay, then there is duplication of the number of DNAs. Okay, so there is duplication. Now there will be cleavage. Okay, the male and the female pronucleus will go on the either side, and now there will be starting of division of the whole of the single cell. Now it becomes 2 cell, 4 cell. We'll talk about that. Okay.
Mosaïque.
Yeah, it becomes. It is called as cleavage, where the single cell will start dividing into multiple cells. Okay. So before we talk about that cleavage, so what are the effects of fertilization? Okay. What are the changes which have taken place because of the fertilization? One is, as we have seen, because of fertilization, there is completion of the second meiotic division in case of the ovum. That is the final maturation of the ovum. Okay. The second is restoration of the diploid number of chromosomes. They were haploid. Initially, half of the chromosome. With the fertilization, there is diploid number of chromosomes. Double. Okay. Or the normal. Okay. The third is determination of the chromosomal sex. How do you determine the sex? Okay. XY. Okay. So if you see here, these are the ovumes and these are the sperms. Ovum will have the X chromosome. Okay. All will have a single, because this is haploid. Initially, before they fuse, they are haploid, so there will be single X chromosome. In case of the male, half of the chromosomes are X and half of the chromosomes are Y. Okay. So if this is the ovum, if it is fertilized by a chromosome having X chromosome, then it becomes female. If a chromosome having Y chromosome fertilize the egg, it becomes XY, that is the male. Okay. So it is mainly the role of the male which will determine the sex of the chromosome. Usually, in traditional countries, in our countries and other countries, usually they blame the female if there is a female born or newborn. Actually, it is the role of if after God somebody else is there, it is the male who has a role to determine the sex of the chromosome, the newborn, male or female. Because half of the chromosomes are X and half of the chromosomes are Y. So determination of sex is already determined. Even if we don't know, but we know that there is fertilization taking place. Okay, we don't know whether it is X chromosome or Y chromosome. But anyhow, already the sex of that zygote. Now, once there is fertilization, it is called as zygote. Okay, so that zygote has already a sex. Either it is XY or XX, we don't know. But it is already determined with the fertilization. Okay. Then determination of polarity of the embryo. What do you mean by pole? Like North Pole and South Pole. Similarly, we have the head and the tail end. Okay. They call it as tail end. Even though the humans don't have tail, but they call it as tail. Okay. The head, they determine the side. There is a part which is determined as head, a part which is determined as the tail. Okay. It's already going on, even though nobody knows. Okay. So it is already determined. Then the initial stages of cleavage. As I said, cleavage is nothing but the division of these cells. It is now a single cell. Now it starts dividing into multiple cells. This is called as cleavage. So once there is fertilization, so it is already ready for the cleavage. From one cell, we have become a whole human being of millions of cells, right? So this is by the process called as cleavage. Okay, so it will initiate at this stage. So these are some of the effects of fertilization. Yeah.
What's polarity? Determination of polarity.
Polarity, that's what I said. One end of the embryo will become the head, one of the end of the embryo becomes the tail. Okay, so that is called as poles. Okay, so it will be determined. We'll talk about that in the second week and third week, how the head and the tail end will be determined. Okay, initially this is the stage when it is already determined. Okay, these are some of the effects of fertilization. One is completion of the second meiotic division of the female ovum. The second is the restoration of the diploid number of chromosome. Third is the determination of the sex. Fourth is the determination of the pole. And the fifth is this will help in the initiation of the cleavage. Okay. So what is this cleavage? As I said, it is division of the single cell into multiple cells. Okay. So it is a process of repeated segmentation of the zygote. As I said, now it is called as zygote. Once the male and female pronucleus are there inside a single cell, now it is called as zygote. So this cleavage is nothing but the repeated segmentation of this zygote, single cell into multiple cells. If you see here in this picture, so this is a single cell with the male and female pronucleus. Now they start dividing into two cells. There are 4 cells, there are multiple cells. Okay. And this will be taking place still with the covering of the zona pellucida. This you should remember, very important. There is still that barrier, thick barrier. Okay. Once the sperm has entered the barrier, now there is formation of barrier. Now the barrier is intact. So you should remember that the zona pellucida, which was a red line before. If you see this picture, you have already seen that zona pellucida. So this is the zona pellucida. This membrane is intact. You should remember this, and you should concentrate on when it disappears. Okay. Later, at one particular stage, it will disappear. So keep on noting when it will disappear. Okay. So now the cell start repeated segmentation of the zygote, and within the barrier that is called as the zona pellucida, and by a method called as mitotic division. Not meiosis, but the mitotic division. Okay, so this is called as cleavage, this process of repeated segmentation of the zygote. Okay. And this gives rise to smaller cells called as the blastomeres. These cells which are formed here, these are called as blastomeres. Here, this is the electron microscopic picture again showing you the cells. These are single cell has become 2 cells, now it has become 4. And there will be 8. Then 12, then 16, like that. Okay. So these smaller cells, these are called as blastomeres. These smaller cells, which are coming from the cleavage, these are called as blastomeres. Okay. Now, when you see the zygote, it is the largest cell in the body. It is a huge cell. Even though it cannot be seen by naked eyes, but it's still, if you compare with the normal cells of the body, there it is a very large cell. Okay. With this division, what happens? The normal size of the cell is restored. The continuous division, the large cell, which was one single cell, now because of division, the normal size of this, because of the continuous division, segmentation, this normal size of this blastomeres or the cells will come to normal size. Okay. Because of the reduction of the cytoplasm. Okay. And these cleavages are of 2 types. One is called as the holoblastic, and the second one is called as the neoplastic. Holoblastic, where the whole cell is divided. Whole cell is divided into 2 parts, then 4 parts, then 8 parts. This is called as holoblastic, because the whole cell is becoming divided. In case of the neoplastic, only a part of the cell becomes a separate cell. Okay, sometime it takes place. Not here, at least. In some other part, there will be small part which becomes a separate cell. Okay, so that is called as neoplastic, where equal division is not there. Here, if you see here, there is equal division of both the cells. But in case of neoplastic, only a part of the cell becomes a separate cell. Okay, that is called as neoplastic. But here, the holoplastic type of cleavage will be taking place. Okay. So these are the different stages by which it will be divided. The single cell will become 2-cell stage after day 1. Okay, after fertilization. We will start counting the days from the day of fertilization. Okay, up to 40 weeks. The whole duration is of 9 months. Duration starting from the day of fertilization. So the day of fertilization is the 0 day. 0 day. Okay. From there, if you start counting, that is the day 1, 2, 3, 4, 5, like that. Okay. So the 2-cell stage will become by the first day after fertilization. The next day becomes the day 1, and it will be 2-cell stage. Then it becomes 4-cell. These 2 will become 4. Okay. Then by the second day. And it becomes then 8, then 12 by the 3rd day, and it becomes finally a stage called as morula, where it is 16 cell stage by the 4th day. Okay, by the 4th day, it becomes something called as marula. It is called as marula because it looks like a mulberry. Okay, so that's why they look like this. Okay, so that's why it is called as marula. Okay, so now you can see here the division. Okay.
It's only 4 stages. Only 4 days.
Yeah, initial stages of division. It will keep on getting cleavage.
5, 6, 7.
Yeah. Continuously it will be taking place. But this stage is very important. That's why we stop at that particular stage. Okay. So if you see here, once again, see the cleavage. So there is the holoplastic. So equal number of size and division is taking place. Here also you can see here 2 cell. Now it has become 4 cell. Single cell. It becomes 2 cell. Then again there is cleavage by that, it becomes 4, then it becomes 8, then 12, then 16, okay, then it keeps on going on, okay, it doesn't stop there. But this stage, 16 cell stage is a very important stage because it looks like a mulberry tree, that's what is called as marula, okay, so this is the stage here. Okay, so the fertilization has taken place in the ampullary part. So here is the ovum release, the first sperm has come, there is fertilization. Now as the cleavage is taking place, this is here is the male pronucleus here and the female, okay, so both the sperm and now there is cleavage, say 2 cell, then 4 cell, 8 cell, 16 cell, okay. During this process, the sperm, the zygote is moving. It is not in the same place in the ampullary part, but it is moving toward the uterus. Uterine cavity. Okay, it is dividing into multiple cells. At the same time, it is moving towards the uterine cavity to get implanted here. So this is called as implantation, where it finally get implanted. Before that, it will be moving, dividing as well as moving. Okay. So now, as I said, the marula is the 16 cell stage, where it looks like a mulberry. That's what is called as marula, and this is by the 4th day, 3rd or 4th day usually. And here all the cells will be of the same size and structure. If you see here, this is the marula, 16 cells will be there, and all the cells will be of equal size and shape. Equal size, shape and everything. Okay. Pardon? Okay. So this is equal cells stage. Okay. Now what happens? Now there is differentiation of these cells. So there is a 2 cell stage, then become 4, then 8 and 12 and 16. This is a 16 cell stage covered by the zona pellucida. This is the zona pellucida, the membrane is still there. Now what happens in this marula stage? The cells which are of equal size and shape, now they start differentiating into 2 types of cells. Okay, one is called as the outer cell mass. Now they differentiate into outer covering cells, they are called as outer cell mass, and the inner cells, this is called as the inner cell mass. Initially, all the cells, 16 cells, will be of the same size and shape. Okay, now they start differentiating into outer cells, which is called as the outer layer of cells. Okay, and the inner cell mass. This is the outer cell mass, and this is the inner cell mass. What is the importance? Why they are differentiating? Because the outer cell mass will later form something called as the trophoblast, which are mainly protective as well as giving nutrition to the embryo. Okay. Then what happens to the inner cell mass? This inner cell mass is the actual embryo, which will develop into actual embryo. Okay. So at the stage of marula, all size of the cells, 16 cells will be of equal size and shape. Then they start differentiating into outer cells, which is called as outer cell mass, which will be mainly protective as well as nutritive. And the cells in the inside, they become separated into inner cell mass, which is called as embryoblast, which will later form the embryo, actual embryo. Okay, did you get it? Okay. And you should remember, the whole of this is still covered by the zona pellucida. Still the covering is there. Okay. Now there is no space here. Now slowly, as I said, it is going into the uterine cavity, right? When it is passing into the uterine cavity, here is the marula. Okay. Once it is in the uterine cavity, slowly the fluid in the uterine cavity, I said there are uterine glands. Okay, they start secreting some fluid, and this fluid start entering this part. And now there are small spaces of fluid inside this. Okay, now they all join together to form a bigger area. Okay, so like this. Okay, so small areas will be there which will all join together to form a larger area. Now this is called a blastocyst, the formation of blastocyst. The small areas, small fluid areas which were there, they all join, coalesce. Okay, this is called as the uterine milk. So once this fluid enters, this is nothing but uterine secretion which has entered this morula, and they start occupying some area and they push the embryo to one side, the inner cell mass to one side, which will later form the embryo proper. And there is formation of a large cavity here. This is called as the blastocele. Blastocele. Cele means cavity, so this is called as blastocele, filled with fluid, some fluid, which is called as the uterine milk. And they are pushed to one side, and this stage where we have the outer cells, outer cell mass, which is forming the trophoblast, and the embryoblast to one corner, and there is formation of a blastocele. This is called as blastocyst. It looks like a cyst. Cyst is that cavity in the body, we have many cysts. Okay, so the cyst is that cavity where there is fluid inside. A bag filled with fluid, that is called a cyst. Okay, similarly here in the embryo there is formation of blastocele. So that's why it is called as blastocyst. This is how it looks like actually. This is how. This is the trophoblast cells, outer cell mass, and this is the inner cell mass. And this is the cavity formation. Okay. I know it is very lengthy. If you take so many lectures, that's why we arrange the lectures with some gaps and other things. And you remove all those gaps, SDLs, and you ask somebody to take the lectures there. And this becomes a problem for you. Sitting for 4 hours is very difficult, I know. Okay. Anyhow, okay. So these cells, these are outer cell mass, which become the trophoblast cells, which are protective, and the inner cell mass, this becomes the embryo. Okay. Now, even the pole where this embryo is attached, this is how this is taking place. 1 cell, 2 cell, 4 cell, 16 cell. This is our 16 cell. Then we have this blastocele.
Uterine.
Okay. Then it is coming out of this cavity. We'll talk about that later. Okay. So this blastocyst will be formed at the almost at the 5th day. Okay. And when the blastocyst is formed, there will be almost 107 cells. As you were asking whether it stops at 16 cell stage. No, as I said, it will continue keep on dividing. Okay. By the stage it comes to blastocele, there will be almost 107 cells. Out of these 107 cells, only 8 cells are embryo. Out of the 107 cells, only 8 are embryo. The remaining 99 cells are the trophoblastic protective cells, all the cells which are protecting outer trophoblast. Okay. So among these, where the embryo is attached, this is called as the polar trophoblast. This is the polar trophoblast where the embryo is attached. The remaining part of the trophoblast is called as the neural trophoblast. So the part where the embryo is attached, there are 30 cells which are protecting the embryo. Even though the embryo itself is 8 cells, the protection for that is by 30 cells.
Okay.
Okay. So that is called as the polar trophoblast. And the remaining part is the neural trophoblast. There will be almost 69 cells. Totally 99 cells. Of the 107 cells will be trophoblast. Remaining 8 cells will be embryoblast. Okay. So I think this will be the last one. Okay. Coming to the implantation. The last stage where the embryo will be implanted into the fetus. There are many slides. We'll stop here and we'll continue tomorrow. Tomorrow or day after tomorrow.
I think so.
Even though number of slides are less, but the thing, concept, understanding the concept is more. Maybe day after tomorrow when we have a second lecture, we'll complete this one and then continue the second one. Okay.